Haberlandt was the first to culture fully differentiated single cells, isolated from the leaves of Lamium purpureum, petioles of Eichhornia crassus, glandular hairs of Pulmonarium mollissima, and stamen hairs of Tradescantia in a simple nutrient solution of Knopp's nutrient solution. He did this to verify the concept of cellular totipotency. The cultured cells survived for up to 1 month and also increased in volume but did not divide. He had proposed that cells in the plant body stop growing after acquiring the features required by the entire organism without losing their current potentiality for further growth and are capable of resuming uninterrupted growth on getting suitable stimulus. Haberlandt is known as the father of plant tissue culture.
Wilhelm Knopp, 28 July 1817, 28 January 1891, was a German geochemist and co-founder of modern water culture, hydroponics. Together with Julius von Saccas, he identified nitrogen, phosphorus, sulfur, potassium, calcium, magnesium, and iron as essential elements of plant nutrition. He developed a nutrient solution known as Knopp's solution. In 1932, White started his famous work on isolated root culture, and in 1934, he announced the establishment of continuously growing root cultures of tomato. Some of these root cultures were maintained by periodic subcultures until shortly before his death in 1968 in India. The medium initially used by White contained inorganic salts, yeast extract, and sugar. Yeast extract was later replaced with the 3B vitamins, namely pyridoxine, thiamine, and nicotinic acid. This heralded the first synthetic medium, which was widely used as basal medium for a variety of cell and tissue cultures. 1939. Gautheret, Nobocour, and White independently established continuously growing tissue cultures. 1941. Van Overbeek introduced coconut water as a medium constituent by demonstrating its beneficial effect on in vitro development of immature embryos and callus formation in Datura. 1946. Ball succeeded in raising whole plants from excised shoot tips of Lupinus and Tropaeolum. In 1947, Braun proposed the concept of tumor-inducing principal tip of Agrobacterium tumefaciens responsible for autonomous growth of crown gall tissue. In 1952, Morel Martin developed the technique of meristem culture of Dahlia to raise virus-free plants from infected individual. In 1954, Muir et al. Succeeded in inducing divisions in mechanically isolated single cells cultured in the presence of inert tissue. In 1955, Miller et al. Discovered the first cytokinin, quinquetin, from autoclave herring sperm DNA. Herring is a fish. From its sperm, DNA is isolated, which is sterilized using steam at high pressure, which is called autoclave herring sperm DNA. From this, kinetin was isolated, which has ability to induce cell division. In 1957, Skoog and Miller put forth the concept of chemical control of organogenesis, root and shoot differentiation, by manipulating the relative concentrations of auxin and kinetine. In 1958, Steward, Usay, and Renert, Germany, independently reported the formation of embryos by the somatic cells of carrot, somatic embryogenesis. In 1960, Jones et al. Successfully cultured isolated single cells using conditioned medium in a microchamber. In 1960, Morel described a method for rapid in vitro clonal propagation of orchids, micropropagation, cocking in 1960 isolated plant protoplasts enzymatically. In 1962, Kanta et al. Developed the technique of in vitro pollination, viable seed formation by in vitro pollination of naked ovules. Murashige Skoog formulated the most widely used plant tissue culture medium in 1962. In 1964, Guha and Maheshwari produced the first androgenic haploid plants of Datura by antherculture. In 1965, Jory and Bojwani demonstrated the totipotency of triploid endosperm cells. Vasil and Hildebrand achieved regeneration of full plants starting from isolated single cells of tobacco in 1965. In 1971, Heinz and Me reported somoclonal variation in the regenerants from callus cultures of sugarcane. In 1973, Nag and Street succeeded in regeneration of plants from carrot cells frozen in liquid nitrogen 196 CE. Ko et al. And Whalen et al. Introduced PEG as a versatile chemical for the fusion of plant protoplastes in 1974. Also in 1974, Zainin et al. identified thioplasmid as the causative factor of Agrobacterium tumefaciens for crown gall formation. NEU 1971. Takeby et al. Achieved plant regeneration from isolated protoplasts of tobacco. 1972. Carlson et al. Produced the first somatic hybrids by the fusion of isolated protoplasts of Nicotian of isolated Procyon, Glauca, and Langsdorff et. 1973. Neitsch and Nouriel succeeded in producing haploid plants from isolated microspore cultures of tobacco. In 1977, Chilton et al. Demonstrated that only a part of the thioplasmid of Agrobacterium tumefaciens is responsible for crown gall formation. In 1984, Horsch et al. Produced the first transgenic plants of tobacco by co-culture of leaf discs with Agrobacterium tumefaciens. In 1986, Abel et al. Produced the first transgenic plants with useful agronomic traits. And in 1987, Sanford et al. Invented the biolistic method of direct gene transfer into plant cells.